All right. So next up, we have Taraman. If you want to come to this stage, Taraman is in the College of Pharmacy in the Department of Pharmaceutical Sciences. Take it away.
Thank you, Jessica. Hello, everyone. Today, I'm here to talk about the story of anti-cancer agent called TK-006, Taraman Kadayat, my name, and 006. Okay. Yeah. As a postdoc, if we want to have a good job, we should be very strong. I mean, we should be very productive. We should have good publication, good track record, and then we'll go in the market, right? Very similar to that, small molecule. If it wants to develop as a medicine, it should have a very good properties. Like it should be very strong, stable in the blood, and it should be very safe. It shouldn't have any side effect. So as a medicinal chemist or drug discovery chemist, my goal is to develop or prepare any molecule that is very safe, effective, and stable. And drug discovery process is a very challenging and lengthy, very complicated process. But in academia, usually what we do that we find the molecule that is very safe, strong, and stable in the mouse model or animal model. And then it goes to the clinical trial, and finally it goes to the market or clinic that can save the thousands of people of life. But people say that this, like a drug discovery chemistry, is a game of whack-a-mole, because whenever we fix one problem, like when we improve the potency of the drug, we have a side effect, or our drug may be unstable. So it's like a game. But my goal is that I want to have a chemical, a small molecule that is very safe, strong, and stable. Okay, I'll repeat it again. I want a molecule that is very strong, safe, and stable. Yeah. And now for that, first we need to find out the disease target, either enzyme or receptor, for example. So in cancer cells, usually there are bad proteins. The bad proteins are degraded by proteasome, right? But to degrade this protein by proteasome wants to be tagged this protein by a small protein called ubiquitin. It's like a magic protein. When it gets tagged to the bad protein, then proteasome will degrade this protein. Okay, and so that the cancer cell will grow and survive, right? But there is a medicine called bortezomib, which is very effective in blood cancer. But the problem of this medicine is that it blocks the 80% of the good and bad proteins. That means it has high toxicity. So there is other very similar magic protein called INAD. When it is tagged to any specific protein, then protein becomes activated and it degrades the bad protein. So if we inhibit this E1 enzyme, then by this drug, which is in the clinical trial, then it shows that toxicity, because it inhibits the degradation of good and bad proteins, around 20% of good and bad proteins, which is not good. So our idea is that we want to have a molecule which selectively degrades this E3, which selectively inhibits E3 enzyme, because this is highly amplified in cancer. So if we inhibit this molecule, we will have safer and more effective selective medicines. But we don't have molecule. So we need molecule. For that, we did high throughput screening. We screened more than 600,000 molecules and found the first hit. We call it hit compound. It has good properties such as like solubility, permeability, and it is not killing our normal cells. That is good. But problem with this drug is that it's weak. It is not strong. So we want to have a strong compound. For that, we took the picture of this compound with protein, DCN protein, UBC12 protein, and we found that there are 5 different pockets that this protein binds. So among them, only 3 proteins are interacting. This is leucine and urea and style pocket is not interacting. So we have opportunity to modify that protein. So we wanted to have a strong compound. For that, we synthesize more than 100 molecules in around 1 year. And then from there, we found that very strong compound. So we became very happy, because now we have very strong. You can see that more than 100-fold improvement in the potency. We're very happy to find this molecule. But when we gave this molecule to the mouse, in 20 minutes in the blood, it became degraded. That means it is not stable. Our molecule is not stable. So we're sad again. Then we wanted to change this molecule. We wanted to change this molecule again. We wanted to replace these different parts of this molecule so that we'll have a strong and stable molecule. So we did. So we found that now we have a strong and very stable molecule because it can stay at least 5 hours in the blood. So we are again happy, very happy, because now we have a strong and like a stable compound, stable molecule. But when we tested this molecule, we found that it has cardiotoxic effect. So it has side effect. It has off-target effect. So that is very bad, right? We cannot have any molecule that is not safe for our bodies. So we became again sad. Then we thought to modify it again. This was our hero because this was very, very potent, stable. Problem is that this is not safe. So we modified this molecule, and what we found that new molecule, and this is like a 15-fold decrease in cardio. This is safer than this one, stable. But the problem is that this is not strong like this. So we lost the potency of this molecule. So we're happy because our molecule was not very toxic, but we are sad because we lost our property, our potency. So we wanted to synthesize more like the better molecule than this. And we found, before that, we wanted to see that why this molecule, they look similar, but why this is not active, why this is not strong. And then we took the picture, X-ray picture of this molecule with the enzyme. We found that this benzyl group, when it had two friends here like this, then it was happy to stay here. But we modified this structure. There was a new group here, sulfonamide group, isotoluene group, as compared to these two. So this benzyl group was sad. He didn't want to these two. So it flipped its position. She said that I want to go to other side. So that means in this position, the benzyl group was interacting here with the leucine pocket, interacting with that pocket. But here, there's no interaction because our benzyl group flipped towards other side. So I told this benzyl group to come back so that we can have a strong, safe, and effective and stable compound. But it didn't. So I thought, okay, if this benzyl group will not come back, then I will introduce new friend here. So in this position, you can see here I introduced 2 carbon bridge system here, and after introducing, we introduced the strength of this molecule because it was almost like 3-fold improvement in the potency. Not only that, the good thing is that this is much safer than these medicines and stable also. So we took the picture of this molecule again, and we found that this molecule very nicely interacting with the enzyme. So in conclusion, starting from the heat molecule, this heat first molecule, which was weaker, I made the strong and stable, stronger but unstable. Then I modified the structure, and then I got the strong and stable compound, but it was toxic. Finally, changing again this structure, I got the strong, stable, and safer compound. So in mouse, we already tested this compound, which is our hero. This mouse in the cancer cell, this sick mouse became happy mouse. So we are also very, very happy. And we hope that our new superhero, this is TK-006, will have similar activity or better than this because that is strong, stable, and safer. So with this, I would like to thank my advisor, Dr. Kip Guy, and our collaborator, and our lab members and different colleagues from the different labs of University of Kentucky College of Pharmacy. And most importantly, on behalf of this DCN project, I would like to thank NIH and NCI for funding this project. Thank you.